Hello guys, I am back. We are now on the second week of our online distance learning. So today, we are going to have the Module 2, Lesson 1. In this module, we are going to have the cell membrane and cell transport as our point of discussion. And for the learning competency, we have described and explained the movement of substances in and out of the cell. When you approach your school, you might pass through a gate in a fence that surrounds the school grounds. The fence prevents people who should not be there from entering, and the gate allows students, staff, and parents to enter. Similarly, cells, either prokaryotic or eukaryotic, they have what we call a structure and serves as cell boundaries. All cells are surrounded by a thin flexible barrier known as the cell membrane. If you are going to look at it, many cells also produce a strong supporting layer around the cell membrane known as the cell wall. Most of the molecules in the plasma membrane are lipids. As you can see in the illustration, lipids are large molecules that are composed of glycerol and three fatty acids. If a phosphate group replaces a fatty acid, a phospholipid forms. A phospholipid fats or phospholipid is actually a molecule that has a glycerol backbone. So, the fatty acid and the phosphate containing group, the plasma, is therefore composed of a phospholipid bilayer. Now, I want you to look into this illustration. So we have here the phospholipid, which has the hydrophilic head, and we have the hydrophobic tail. That the process of maintaining balance in an organism's internal environment is what we call homeostasis. Homeostasis is essential in the survival of the cell. Also, homeostasis is also known as equilibrium in our body. Homeostasis is essential in the survival of a cell, and one of the structures that is primarily responsible for homeostasis is what we call the plasma membrane. Why? Because the plasma membrane is responsible in controlling what enters and leaves the cell. So what are the functions of the plasma membrane if you are going to recall it? First, it is a protective barrier. Second, we have to regulate transport in and out of cell. That's why it's called selectively permeable. It allows cell recognition, provide anchoring sites for filaments of cytoskeleton, provide a bonding site for enzymes, interlocking surfaces bind cells together in the junctions, and contains the cytoplasm, which is called the fluid in the cell. Notice in the figure that each phospholipid is diagrammed as a head with two tails. The phosphate group in each phospholipid makes the head polar. The polar head is attracted in water because water also is polar. The two fatty acid tails are nonpolar and are repelled by water. So the phospholipid bilayer, or the plasma membrane rather, makes up the cell membrane two layers. That's the reason why we call it as bilayer. The phosphate head is hydrophilic. So when we say hydrophilic, it's water loving. It is polar with water. While the two tails, it is fatty acid. That's why it's called fatty acid tail. And it is hydrophobic. So water fearing. And the phospholipid makes the membrane selective in everything that crosses the cell membrane. To look closely at the phospholipid bilayer, this is how they are situated in the plasma membrane. So we have here the polar, the hydrophilic head, and if you're going to look at the center, these are the nonpolar or the hydrophobic fatty acid tails. And on the other side again, we have the polar hydrophilic head. So this phospholipid bilayer, this is how they are being situated in the aqueous environment of the cell. So if we're going to look at figure A, so that is now the phospholipid molecule. That is the thing that we've been discussing previously. And then on the figure letter B, we have the phospholipid bilayer now. Okay, that is now the whole semi-permeable membrane, which is the plasma membrane. Now why it's called semi-permeable? It does not allow everything to enter the cell. Only small molecules and hydrophobic molecules would flow easily through. Examples are the oxygen, carbon dioxide, and water. Since the plasma membrane is considered as semi-permeable, the ions, hydrophilic molecules larger than water, and large molecules such as proteins do not move through the membrane on their own. After discussing the structure of the cell membrane, we are now moving on to what we call the cellular transport or the cell transport. Cellular transport moves substances within the cell and moves substances into and out of the cell. Now, if you are going to relate it in our real world, let's try to imagine studying in your room while cookies are baking in the kitchen. You probably did not notice when the cookies were put into the oven because you could not smell. But as the cookies bake, the movement of the aroma from the kitchen to your own room happened through a process what we call diffusion. So we're going to continue our discussion on the different types of cell transport. As you can see here, we have passive and active. So as the aroma of baking cookies makes its way to you, the particles are moving and colliding with each other in the air. This happens because the particles in gases, liquids, and solids are in random motion. Similarly, substances dissolved in water among constantly in random motion is what we call the Brownian movement or the Brownian motion. So this random motion causes what we call diffusion. And that is an example of a passive transport. And passive transport requires no energy, the molecules move from area of high to low concentration. As I said earlier, diffusion is a passive process, which means no energy is used to make the molecules move. They have a natural kinetic energy. It makes down the concentration gradient. So look at the illustration very well. So that is an example of diffusion. Another example of diffusion is if you are going to put or drop a dye in a beaker of water. Now, diffusion causes the dye to move from high dye concentration to low dye concentration, and the colors become very blended in water. So look at the different beaker, from beaker A down to that of beaker C. So the dye is evenly distributed. Why? Because the molecules of dye is now mixed with that of the molecules of water. A very simple example of a simple diffusion is the movement of oxygen or the entry of oxygen or water. The oxygen or water diffusing into a cell and carbon dioxide diffusing out. So look at the illustration very well. So the materials move down their concentration gradient through what we call the phospholipid bilayer. In addition to water, cells need certain ions and small molecules such as chloride ions and sugars to perform cellular functions. As we have seen earlier from the previous slides, water can diffuse across the plasma membrane, but most of their substances cannot. Another form of transport is what we call facilitated diffusion. Still, facilitated diffusion does not require energy. It uses transport proteins to move from high to low concentration. Look at the plasma membrane in the illustration. There is what we call a protein. So that protein helps in facilitating the entry of the different ions. So examples are the glucose or amino acids moving from blood into a cell. So the passage of materials is aided both by a concentration gradient and by a transport protein. Another illustration for you to take note how facilitated diffusion occurs in the plasma membrane. So look at the ions that enters the cell. So from outside of the cell to the inside of the cell. Another thing that you need to remember when you speak of diffusion, there is what we call the process of osmosis. Osmosis is the diffusion of water across a membrane. So look very well into the animation that between there is what we call a semi-permeable membrane. Now, in osmosis, my dear students, there is a diffusion across a membrane. It moves from high water potential. So when we say high water potential, there is low solute to low water potential in which there is high solute. Diffusion of water through a selectively permeable membrane is also illustrated in this figure. So as you can see, the small circles there are what we call the water molecules. And the big circles there, we have the solutes. So earlier, we have said from high potential of water, okay, to low potential of water. So that is the movement, or that is the diffusion of water through a selectively permeable membrane. So for now, we're going to consider the different types of environment of the cell. So we have here the isotonic, hypotonic, and hypertonic solutions. So let's define first the different terms. So when we say isotonic, a solution of equal concentration to a cell, and when we say hypotonic, a solution of lower concentration than a cell in which there is higher water potential. While hypertonic, a solution of higher concentration than a cell, meaning to say there is lower water potential. Note, my dear students, that using these terms always involve comparison between two different solutions. So let's look into different illustrations here. So we have here the cell in isotonic solution, the cell in hypotonic solution, and the cell in hypertonic solution. So let's take, for example, that the cell is being soaked in a salt solution, a 10% salt solution. So whether it's outside then its environment or its environment within the cell. So when we say solution or the cell in a solution of isotonic, as you can see, the amount of the salt are the same. So in its environment, there is 10% salt solution, and within the cell, there is 10% salt solution. So if you are going to look at it, since the amount of the solute and the amount of the solvent are equal, water therefore goes in both directions. While if the solution is in hypotonic state, when we say hypotonic state, the amount of solute in the environment is less than that of the amount of solute in the cell. So in the environment, we have 10% solution of salt, while within the cell, it has 20% salt. Now, so how the water moves into the cell? So the water moves, therefore, into the cell. Why? Because there is greater amount of water outside the environment. While on the third illustration, the hypertonic solution, as you can see, the amount of salt in the environment is higher than that of the amount of salt in the cell. So, water outside is less amount, while the water inside the cell is greater. Therefore, water moves out of the cell. So, that's how we could illustrate the different environments of the cell. So, for now, we're going to compare the cells, the animal cells, and the plant cell in the different solutions. Our point of reference will be the isotonic solution. Looking at the isotonic solution, the cell is at equilibrium with the solution, and there is what we call a net movement of water. So look at the animal cell. The cell retains its normal shape. Same is true with that of the plant cell. It is in their normal state. So most cells in organisms are actually in isotonic solution. Another example of this is the blood. Our blood is in isotonic solution. So let's look the animal cell in that of the hypotonic solution. Look at the hypotonic solution. So the animal cell, the tendency, if there is more water that enters the cell, the tendency it will burst. And what about the plant cell? So the plant cell, since the water stays in the vacuole, look at the vacuole of the plant cell in the hypotonic solution. So it's still turgid or it's still normal. Why? Because the plant cell does not burst. The plant cell does not burst because there is what we call a cell wall. While in that of the hypertonic solution, look at the animal cell in the hypertonic solution. The tendency is that it will shrivel. Why? Because most of its water will get out from the cell. And look at the plant cell. Look at its vacuole. It will also plasmolyze. So those are the differences between the animal cell and plant cell if they are being placed in the different environment. The hypertonic solution, isotonic solution, and hypertonic solution. Sometimes substances move from a region of lower concentration to a region of higher concentration against the passive movement from higher to lower concentration. This movement of substances across the plasma membrane against a concentration gradient requires what we call an energy or ATP, and this is called active transport. In the illustration, you could see how active transport occurs with the aid of what we call the carrier proteins. So the molecules again move through a transport protein, but now energy must be expended to move them against their concentration gradient. In this illustration, guys, you could see how active transport occurs. One common active transport form is called the sodium-potassium ATPase pump. This pump is found in the plasma membrane of animal cells. The pump maintains the level of sodium ions and the potassium ions inside and outside of the cell. This protein pump is an enzyme that catalyzes the breakdown of energy-storing molecule. The pump uses the energy to transport 3 sodium ions out of the cell while moving 2 potassium ions into the cell. The high level of sodium on the outside of the cell creates a concentration gradient. Look into the illustration and follow the different steps to see the action of the sodium-potassium ATPase pump. How are the large particles are being transported? Some substances are too large to move through what we call the plasma membrane by diffusion or transport proteins and must get inside the cell by different processes. There are 2 processes that you will need to take note. First, we have the endocytosis. So when we say endocytosis, it is the process by which a cell surrounds a substance in the outside environment, enclosing the substance in a portion of the plasma membrane. The membrane then pinches off and leaves the substance inside the cell. The substance that is shown in the figure, it is actually being engulfed and enclosed by a portion of a cell's membrane. The membrane then pinches off inside of the cell, and the resulting vacuole with its contents moves to the inside of the cell. Let's look into this process in endocytosis. There is what we call the pinocytosis. So when we say pinocytosis, it is called cell drinking, in which it involves liquids. And we have also phagocytosis, the cell eating, which involves solid. Another type of the transport of large particle is what we call the exocytosis. Exocytosis is the secretion of materials at the plasma membrane. The illustration, as you can see in the presentation, shows that exocytosis is the reverse of endocytosis. Cells use exocytosis to expel waste and to secrete substances such as hormones produced by the cell. Sum up the cellular transport, there are two kinds. We have the passive and the active transport. Look at the illustration very well in the presentation, guys. Look into the movement of the substances in the passive transport and compare it to the active transport. So in the passive transport, always remember that the particle moves in an area of high concentration through a protein. And that of the active transport, energy from ATP is used to move a particle through a protein against the direction of diffusion. So that's all for today, guys. Bye till next time.